Hello, everyone. My name is Ariella Beverly. I am a first year IBS PhD fellow. And for my second lecture, my asynchronous lecture, I will be talking about the pathogenicity of bacteria. The three objectives I want everyone to keep in mind for this lecture is by the end of this PowerPoint, what I want you to be able to do is understand how bacteria enter host cells. We want to be able to detail how pathogens penetrate or evade host defenses. And then I also want you to be able to explain how pathogenic bacteria damage host cells. So, pathogenicity is the ability of something to cause disease. For bacteria, in order for them to be pathogenic, they have to gain access to the host. And this is done various ways. Bacteria is ubiquitous, meaning that it's everywhere. It can be found anywhere and everywhere unless we purposely exclude them from that environment. So, a lot of the ways that we interact with bacteria, pathogenic bacteria, is through the air. They can be transferred through water droplets. We can They can come from the soil. They can be contaminated on our food. Um, Animals can host or yeah, animals can be infected with pathogenic bacteria. Those are called zoonotic infections, bacterial or viral infections that happen from an animal host and are transferred then to a human host. They can be on the plants that we interact with, and we also carry them. There are billions upon trillions of bacteria in our gastrointestinal tract that can be opportunistic if given the chance. And that is typically through like a fecal oral transmission route. Most bacteria are going to gain access to the inside of, to human hosts by respiratory and gastrointestinal tracts. But it can happen through several different it can happen through seven different ways. Um, so one way is through the contact with mucous membranes. So we're talking about that soft, moist lining that you will see in your stomach or in, the vaginal tract, the respiratory tract, the gastrointestinal tract, the oral tract, um, with the urinary tract. And again, microbes can be easily the microbes can easily access a host by inhaling through the nose or the mouth. We can uh, get an infection through the consumption of contaminated foods or waters. If you didn't wash your hands properly and you make contact with your mouth or your face or your eyes, that's a very easy way to contract bacterial infections. So the skin is the largest organ on our body, and it's one of our first lines of defense. Um, it's usually really great at keeping bacteria on the outside, but when we have issues like broken skin, abrasions, that can give bacteria the exact access that they need. So the first mechanism we're going to talk about is bacterial adherence. So when we're talking about bacterial adherence, we're just talking to bacteria sticking to a host cell. They can't just free float around the body, right? They would typically attach themselves to a cell surface. They do this through fimbriae, which are like these long kind of, I like to think of them as like a fish hook, right? So it's kind of just floating out there, waiting to be anchored on something. And then you also have adhesions. So we have two mechanisms, fimbriae and adhesins. Another way that bacteria will stick inside a host cell is the creation of biofilms. And this is just It's a communal way to protect the bacteria. So let's say a bacteria gets access to you, a group of bacteria gets access to you. Everybody's floating around trying to find the perfect place to land. Everybody's floating around trying to figure out where is the best place to live? Where is the best real estate? Where are the good neighborhoods at with good food, the good resources, plenty of space? Once one bacteria figures out a place that everybody can kind of stay, then that bacteria will adhere to that surface and then it will release um, proteins, or we call it quorum sensing. And I use the analogy a lot of, you finding a good place to eat, or you find in a store with a really, really good sale, and you call up your friends and you're like, you will not believe the restaurant I just ate at. You will not believe the sale that TJ Maxx is having right now. You need to hurry up and get over here. And it's the signal for all the other bacteria in the body to come congregate in this area. And this happens because when bacteria create these communities, they create a very intricate network that actually protects them and increases their chances of survivability. The next mechanism I want to talk about is avoiding host defenses. So the bacteria have been able to get into your body, right? How does it stay there? How does it make a home? How does it survive or even thrive in the optimal conditions that your body may provide for it without being detected by your immune system? So the first way, or the first discussion we're going to have is about capsules. So capsules are this extra layer that coats the bacterial cell wall, and they can actually resist host defenses by impairing phagocytosis. Imagine this is the way I think about it: picking up something that you think is supposed to be soft when you go to eat it, and it's actually hard and bitter and nasty. And so you spit it out. You don't want to eat it. That's kind of the way I understand phagocytosis and the capsule's impairment of it. Um, it's also really good at concealing some of those subcapsular antigens. So, bacteria, are so very interesting. Bacteria have kind of like telltale, telltale surface proteins. Okay. And our bodies have grown through adaptive immunity, they've grown to recognize them. But capsules, right, that extra layer on the outside of the bacterial cell actually prevents the recognition of those cell surface proteins, and that helps it evade the immune system. So opsonization is a really cool immune response, which happens when antibodies are recognizing a bacterial cell or just a foreign body in general, and they will tag it with a complement, and that will kind of mark it for phagocytosis. And that's what happens normally, right? Now, to jump back to bacterial adherence for a moment, when when those bacterial cells make that contact with the surface cell proteins of the host cell, what will typically happen is that the host cell will then engulf the bacterial cell. Now, this isn't the same thing as phagocytosis by neutrophils, right? Because um, that mechanism will end up in the degradation and the ultimate death of the bacterial cell. What happens here is that when the bacterial cell sticks to the surface cell proteins, the surface cell receptors on the host cell, and then the host cell engulfs it, it can actually stay inside the cell. There are several mechanisms, one in which bacterial cells can kind of just hide out while, essentially while the cops are looking for them, right? While the immune system is looking for those foreign bodies, those intruders. And then when it senses that the body is no longer under that stress, it'll exit the cell. It can also hop from cell to cell um, using the manipulation of some of those active filaments that we've seen. Now, the other mechanism is that these bacterial cells can actually live and thrive inside the host cell. It'll use up the nutrients, it'll use up the space, it'll be a terrible, rude, unwelcome neighbor and it'll even replicate until there's no more space, there's no more resources inside the cell for it to use. And that's when you wind up with host cell lysis, the death of the host cells because it's just too much. It's too much space being taken up. And then when the cell dies, when the cell does lyse, you have all of these new generations of bacteria that are then able to spread throughout the body. Um, there is there are also some enzymes that bacteria can express that'll aid in hiding from the host cell immunity. One such case, Staphylococcus aureus is able to produce a coagulase. And what this enzyme is going to do is turn fibrinogen, which is like the precursor kind of dormant version of fibrin, into the active version. So turning fibrinogen into fibrin. What that results in is the cells clotting together, right? It's called agglutination. It becomes a big conglomerate, a big clot with the bacterial cell at the center. This provides a very, very clever cover and concealment from some of those neutrophils. Think of it like um, jumping in bushes, right? Hiding out in a building, um, taking a bunch of blankets and throwing it over yourself. It's a disguise, a very clever disguise, because it's using host cell components to be able to disguise itself from host cell immunity. And then we also have antigenic variation. That's a big word for Elmo, but listen, all this means is the bacterial cell is just is changing its disguise a little bit. I don't know if you remember earlier, but earlier we talked about how some of those surface cell proteins on bacteria make it really, really easy to identify um, by host cell immunity, right? So it's like showing up to work in the same jacket over and over and over again. People know when they see a red coat out in the parking lot, hey, that's Jim. Jim shows up to work in the same red coat. He's been showing up to work in the same red coat for the last 3 years. Okay. We know that's Jim out in the parking lot before Jim even walks in the building. Kind of the same thing. There are repeated proteins on the surface of bacterial cells that will kind of give it away when if it's caught by the host immune system. So what can happen is that you can have slight variations, right? You can have the altered expression of just a single protein as seen in this diagram here. Um, and that can completely make it unrecognizable to an antibody, right? Now that antibody can't bind to the antigen on that receptor because you changed just one protein. You can also alter several proteins, um, which can change the target. It can change the amount of the target, right? So if there's a certain threshold, that's a certain threshold of residues that needs to be met for the antibody to bind to it, you can switch those things so that the antibody is no longer able to identify the antigen. And remember, in opsonization is being found out, right? It's being caught. Um, Antibodies will aggregate on the cell surface or on the surface of a foreign body. It'll congregate, right? It's a bunch of attention that you as a pathogen don't want, because it marks it for phagocytosis. It marks it effectively, it marks it for death, right? So if you can alter just a bit of those proteins, then you can aid yourself in your survivability rate. You can enhance um, or improve your chances of surviving in a host cell. Um, I know we've talked about phagocytosis a couple of times, but it really is a very, very clever way for bacteria to hide from their host. Just is what it is. Again, once bacteria are able to bind to those surface proteins will actually change the conformation of some of those structural proteins, some of those structural components in the cell membrane, and allow the cell to be engulfed inside of the host. And again, this isn't for degradation, right? This is, by definition, it is phagocytosis, but it's not phagocytosis with an end goal of degradation. These cells can remain, these bacteria can remain inside the host cell. And they can use up resources. They can reproduce until there's no room left and the cell bursts, or they can move from host cell to host cell if it's a sort of, hiding from the cops scenario. And I know we've talked about biofilms, but we're going to talk about them again. Um, when bacteria are able to gather in these communities and create these really interesting infrastructures, these very intricate protections, they become so much more pathogenic. Does that make sense? You can jump me. If it's just me and you on the street, it's me. Well, let me not say that. If it's me against 20 of y'all, you can definitely jump me. But if it's me and mines and I roll 50 deep against you and yours and it's only 20, and we're bringing reinforcements and we got getaway cars, now the scales have been tipped, right? Everything is better in community. Everything is better with a buddy. If you didn't like the fighting scenario, think of it this way: all my ladies that have been out with your girl, y'all know we don't go to the bathroom without one another. We are not leaving with one another. I don't care if you just met a cute man at the bar. We are leaving together because there is safety in numbers. You see that all throughout nature, and bacteria, it's no different. There is safety in numbers. So the last thing we're going to talk about are the mechanisms of host cell damage. So now the or excuse me, the bacteria has successfully infected the host, right? We've successfully um, gained access to the host. We have been able to evade the immune system. And now we're going to talk about some of the mechanisms of host cell damage. So there are some direct damage ways. We've already talked about them. Cell lysis, I won't continue to harp on that. I think you guys got the concept with that. Um, but this is a way to affect direct damage to a host cell. All right, so we also have the production of toxins. There are two types of toxins that I'm going to talk about. The first is exotoxins, and the second is endotoxins. Now, before we get into the definition, this is going to get slightly confusing. I know what you're thinking. Endotoxins, those are inside, and exotoxins are outside. Not this time, buddy. Um, Exotoxins in this case are proteins that are produced inside the bacteria and then are secreted to the outside. We call them exotoxins because we're releasing them outside, okay? And then we have endotoxins. Endotoxins are structural components of the bacterial membrane. And you typically don't see these released until the bacterial cells die. Now, we call those endotoxins because the structures are in the bacterial membrane, okay? So, exotoxins are proteins inside the bacterial cell that are released outside, and endotoxins are structural components of the bacterial membrane that are inside the bacterial membrane, okay? Uh, So a this is an example of an endotoxin. So endotoxins are only found on gram-negative bacteria. Gram-negative bacteria have an extra membrane, okay? And on this extra outer membrane is where you will find your endotoxin. So you have what's called um, lipopolysaccharides or LPS, and a component of LPS is lipid A. Lipid A is going to be your endotoxin. Does that make sense? So you have um, uh, LPS, which only occurs on gram-negative bacteria because it has the extra layer, and then component of LPS is going to be lipid A, and that is going to be your endotoxin. An example of an exotoxin would be Shiga toxin that's released by E. coli. So the Shiga toxin that's produced by E. coli will um, be released. So the Shiga toxin is actually two components. And what winds up happening is that when it's in the cell, it's released from that subunit. So in this case, you see it looks like a flower, okay? And the center of the flower is released, and that is actually going to inhibit protein synthesis, okay? And the B subunit is actually going to be recycled by the cell. But that is an example of a exotoxin. Exotoxin. It is produced inside E. coli, but it is released to the extracellular space. One of the last ways, I think this is the last way that I'm going to talk about uh, damage to host cells is through plasmids. So plasmids are these little circular genetic rings, and it's separate from the DNA, right? So in this diagram here, you have this is E. coli, right? And then you have this spaghetti looking mass, that's your DNA. And then you have the spaghettios on the other side. The spaghettios are your plasmids. And what's so crucial, what's so important about those plasmids is not only can they contain antibiotic resistant genes, as this diagram shows, but they are actually what encode for those exotoxins that we were talking about in the last few slides. Um, these are what's going to add virulency to your pathogens. Okay, this is what's going to make them dangerous, and this is what can carry on the pathogenicity or the virulency from generations to generations, or to bacteria of the same generation, because we have two mechanisms of transferring genes. You can do it from parent cell to offspring, or you can release these plasmids and have them be picked up by bacteria of the same generation, which adds to genetic variation, which adds to virulency, which adds to antimicrobial or antibiotic resistance, making pathogenicity a real big problem. So we're going to take a second to review before I ask you my questions. Um, the key points of this lecture. Okay. So we talked about our ports of entry, right? How bacteria can enter through the mucous membranes, through the skin, through cuts, through scrapes and whatnot. We talked about how bacteria adhere to host tissues through adhesins or ligands that bind to the surface cell receptors. And I want you to remember those ligands are on the fimbriae. So we had the adhesions and then we had the fimbriae, right? And those are what's used to stick to tissues inside the cell. And then we also talked about biofilms, right? Those communities that adhere to a surface inside the host. We talked about the evasion of host defenses, how capsules make it really hard for phagocytosis. We talked about how, again, those fimbriae are those adhesive structures that can stick onto cells and help with the encapsulation, the phagocytosis of the bacteria by a host cell that is not a neutrophil. Remember, we talked about phagocytosis for survivability, not phagocytosis for degradation. Two separate things. We talked about how coagulases can create those fibrin clots that kind of hide the bacteria. So when a neutrophil is looking for it, it can't find it. Can't use phagocytosis for degradation. Aha, you're catching on. Great job. We talked about antigenetic variation, which are slight alterations in surface cell receptors that make it harder for antibodies to recognize them and to bind to them. We talked about how bacteria can live and replicate intracellularly after phagocytosis, right? Once they get in there and they see if it's got good living, it's not going to go nowhere. It is not going to go anywhere. And then we again talked about biofilms because biofilms are integral for providing that communal protection from host immunity. Finally, we talked about host cell damage and how endotoxins, remember, endotoxins are the lipid portions of lipopolysaccharides or LPS, and those only are seen in gram-negative bacteria. That's because gram-negative bacteria have that extra layer. You got it, you got it, you got it. So endotoxins are called endotoxins because they stick inside that extra layer on gram-negative bacteria. And exotoxins are proteins that are excreted from the inside of a bacterial cell to the surrounding areas. And then finally, we also talked about plasmids and how plasmids are incredibly important to virulency because they carry some of those resistance genes and those exotoxin genes. Okay? If you have stuck with me this long, thank you very much. I appreciate you listening to this lesson. And then I want to apologize to you because we're going to do some questions. Yes, we're going to do some review questions. All right. So for our first question, I want you to name the two most common ports of entry for bacteria. Can you remember the two most common ports of entry for bacteria? Question number two. Can you tell me what the function of fimbriae is? What is the function of fimbriae? Question number three. It's not really a question. It's more of a fill in the blank. Some bacteria can alter their surface cell what to avoid detection by antibodies? Bacteria can alter their surface cell blank to avoid detection by antibodies. Please fill in the blank. Question number four. This one actually is a question. Are endotoxins on the inside or the outside of the cell? Can you remember? Are endotoxins on the inside or the outside of the cell? And finally, True or false? Plasmids contribute to virulency. Is it true or is it false that plasmids contribute to virulency? Please complete these questions and shoot me an email, um, and I will be waiting for your response. Thank you very, very much for sticking with me. Thank you to my classmates for all of your support. Thank you to my instructors who are always challenging me. And I will see you all next time.